So what I'm going to talk to you tonight is the current state of a large breeding program that's been going on here at the lab for about the past 25 years, looking at bees with varroa-sensitive hygienic behavior. We'll talk about how this stock was developed, how we measure varroa-sensitive hygienic behavior, and what we're doing in collaboration with James specifically, and the rest of the beekeepers up in Michigan that are aligned with this, to develop these stocks for more broad acceptance and use by the bee industry. So yeah, so this is really nice getting a good tag team action here with Cameron, Jack, and I. So Cameron and I have talked a lot about some of these things with varroa mites, and we keep hitting you over the head with these issues on varroa. But I'll tell you what, in the time that I've been a research scientist here at the USDA lab in Baton Rouge, the only thing that kind of makes a lot of sense and that's pretty consistent, no matter where we're at, whether we're talking the Dakotas, down here in Louisiana, or anywhere in the country for real, is that bees do better when they have less varroa. I think that's one thing I can say with a lot of confidence in honeybees. When there's a lot of things that could be not interpreted correctly, that's one thing that is the same no matter where you go. Less varroa, you're going to have healthier bees that produce more honey, have more bees when you need to do pollination, and things like that. And as Cameron was mentioning, there's a lot of different ways that we could keep varroa mites low in colonies, looking at some of the IPM methods, but then also looking a little bit more at the miticide route. And unfortunately, here in the United States, that's mostly what we've been doing for the last 40 years now, is just hit it with miticides. Miticides seem to be the only thing that some people do to keep varroa mites low. And what we really need to start thinking about are varroa management systems. So using a little bit of IPM, a little bit of miticides, but also to looking at genetics in honeybees to be able to provide varroa control on their own. So what I'm going to talk to you tonight is the current state of a large breeding program that's been going on here at the lab for about the past 25 years, looking at bees with varroa-sensitive hygienic behavior. We'll talk about how the stock was developed, how we measure varroa-sensitive hygienic behavior, and what we're doing in collaboration with James specifically, and the rest of the beekeepers up in Michigan that are aligned with this, to develop these stocks for more broad acceptance and use by the bee industry. So with that in mind, I'll get started talking about everything. So selective breeding is agriculture. Those two things are inseparably linked, because when we think about the history of crop development, this has been the result of either historically passive selective breeding or modern times with active selective breeding. So corn, for example, corn didn't always exist as this big lump with knobs all over it. It actually started off as this little tiny grain, these grains on a small little stalk called teosinte. And over time, through domestication through the Mexican highlands, that stalk got bigger with more kernels until we end up with the giant cobs of corn that we have today. And then also selecting for different characteristics on wild brassica crops. These are the mustard family. If you select for different features, you could actually end up with different varieties of that plant that we know today as cabbage, if you're selecting for the terminal buds, or Brussels sprouts, if you're looking for the lateral buds on that plant. And then even if you're selecting for leaves, you could develop kale and cauliflower and broccoli if you're looking for the flowers. So you can see here that the, hold on a second, look at my laser point. You can see here how just selecting for different traits on these plants can get you these different varieties and different improvements. But it's not only plants that you can do this with, it's livestock, it's animals. So, based on what you want to develop in that livestock, you could select for it. So with cattle, you can either select for either dairy cattle or beef cattle. And then within those groups, there are different kinds of within those different regions that you would use them for. Like, for example, you raise Holsteins to make prodigious amounts of milk, but you don't really necessarily raise them for beef production. Then also to Jerseys and Red Poles typically have a lot more higher fat content in their milk that's usually used for butter. Similarly, if you're producing beef, Herefords and Angus are very adaptable to many different conditions, especially down here in the South. They have a consistent product, and they tend to be tolerant of many conditions, especially down here in the South. They can breed them for tolerance and also cross them with things like Brahmins and things like that. That's what a landowner down here does. So you can see here that just based on what you want out of any kind of livestock, you could selectively breed for it and make it ideal. And honeybees are really no different. So over time, while honeybees have been managed, honeybees actually have never been domesticated, believe it or not. They've been managed because swarming honeybees go feral real quick. But what we can see here is that we have selected bees for things that we like, right? Like high honey production. You can see these frames of sealed honey. We could select for color. So like these cordovan queens, you can see these light golden queens. A previous selective breeding program has developed the Minnesota hygienic stock, the ones with these high levels of hygiene to freeze kill brood. You can see that in that figure here in the bottom. And then even specifically for beekeepers who do especially almond pollination, we can select bees that develop, that expand their colony sizes very rapidly. So in February, they have plenty of bees to pollinate almonds. And Rob Page, when he was moving around all over the country, he would consistently select bees for this pollen hoarding behavior. So with honeybees is that they are amenable to selective breeding, and all you have to do is identify the characteristic you want, find a way to measure it, determine if there's a heritable component, and selectively breed for it over time. And so when we're looking at things that have, and that's when we're looking at things that we have artificially selected. However, Mother Nature doesn't have those those goals in mind. Natural selection really only cares about surviving to the age of reproduction, and then everything after that is not necessarily part of the program. So across the country, across the world, actually, there are natural varroa mite surviving honeybee populations. So, and this list needs to be updated because this is already 8 years old. But here in the United States, Tom Seeley has explored the Arnott forest bees that find out that, yes, they do significantly have fewer varroa mites. And Barbara Locke over in Gotland Island up here in Sweden has shown that they could have mites that naturally survive varroa mites. And so there are a lot of different ways that bees can have low varroa mites. So some of the traits with genetic basis for low varroa populations include infested brood removal and suppression of mite reproduction. That we'll talk about those two things as part of varroa sensitive hygiene a little bit later. Social apoptosis. This is where infested pupae self-destruct. So the Russian honeybees, this is one way that they could interrupt mite reproduction. Social apoptosis or pupal self-destruction. There's some European populations that have demonstrated that there's something in the brood. The brood don't die like the social apoptosis, but there's something in that brood. We don't know if it's a chemical. We don't know if it's something that is inside the pupae that the mites feed on and then die. We don't know what it is, but there's something in that brood that kills varroa mites. And this is kind of interesting. Even in the absence of workers, those brood can destroy varroa. So something interesting going on there. Grooming behaviors, whether we're looking at auto grooming where the bees groom themselves or allo grooming where bees groom others, are a way to dislodge varroa and knock them off. One thing that's kind of paired with grooming often, and this is specific in Russian honeybees and then also too in the ankle biters, is that they can result in varroa damage. You can see the varroa with the legs chewed off, and they can no longer hold on to the bees. Small colony size and frequent swarming are typically used a lot in those feral populations that can withstand varroa. So that by doing that, they could leave varroa behind and establish new colonies. While those are two great ways to keep varroa populations low, in feral colonies, there are two characteristics that you definitely don't want if you are a beekeeper who's looking to keep bees for productive production of honey and pollination services. So you can see there's a lot of different ways that we could get resistant bees. And the reason why we're so concerned with this is that varroa are a huge problem with honeybees. And unfortunately, the biggest thing that we do when we have varroa is we treat. And Cameron told you about how we treat with some things like apistan that kind of don't work as much anymore. Fluvalinate doesn't work at all. And so all these products go into research and development with different active ingredients and different modes of application. And it just keeps getting much more expansive. While miticides can be a part of your varroa management program, it shouldn't be your entire varroa management program. Because you know what happens when you keep applying the same thing over and over again, resistance develops. And if you know who I am in my other part of my research career, you would know that I'm doing a lot of research on amatraz resistance and varroa. So we need to kind of get off this whole miticide treadmill in order to provide long-term sustainable solutions to the varroa problem. And so when we're looking at developing resistant stocks of honeybees, we need to make sure that we're talking, distinguishing between resistant and tolerant stocks. So resistant stocks, or resistance in general, could be the ability to keep a parasite population or pathogen levels below the damaging threshold. So this is actively suppressing the parasite or pathogen, whether it's with some kind of biochemical cue, or in the case of honeybees, with the behavior. That is significantly different, though, than tolerance, where tolerance is the ability to maintain some level of health in spite of increasing parasite and pathogen loads. So this could be things like, I saw honeybees with 30 mites per 100 bees and they were doing fine. While that might sound good that they're still surviving those high levels of mites vector viruses, and over the long term, they could act as reservoirs for varroa. So we really don't want to breed for tolerance. What we really want to do is breed for resistance, where the bees are keeping varroa levels actively very, very low. Just do something here real quick. Okay. So again, like I mentioned, this program started about 25 years ago at the lab. So John Harbaugh and Jeff Harris were the ones who started this program, and I'm just kind of carrying that torch right now. And when they were doing the selection for varroa sensitive hygiene, they actually started with only 28 colonies, not very many. Believe it or not, there tends to be enough variation in most honeybee populations where you could select for these characteristics. And like I said, they started with only 28. And so what they were able to find were characteristics that had a high heritable component. If there is no heritable component, you cannot breed from it. The higher the heritable component, the faster you'll make progress in your breeding. And what they found had the highest heritable components were the features of non-reproductive mites, suppression of mite reproduction. You might have also heard it called that as well. Looking at infested brood removal, that hygienic part of this program. And then the proportion of mites in the brood. So when they looked at these things, they found that these are the things that we should be breeding from. And it's really important to identify the heritable characteristics. So this is a figure right from their paper, and I just want to go over this with you really quick. To identify what they found in this population had higher heritable characteristics. So here on the left-hand side, we have the characteristic that they're evaluating, and then they had the range of heritable components to it, right? And then this H square, this is your strength of heritability. 1 meaning it's completely inherited, 0 means there's no heritability at all. The higher the H squared, the higher heritability. And so things with high heritability here are shown as gray. You can see here the suppression of mite reproduction had a heritability score of up to 0.46, which is pretty high. Hygienic behavior had a heritable component of 0.65, again, very high. We're usually looking for an H square of over 0.25 to be heritable. You notice here, physical damage to mites, there was no heritable component to that, 0. If you try to breed something with a 0, you won't make much progress. And then again, the mites per 100 cells of brood, highly heritable. One thing I really want to point out here, though, is look at this last one, mites per 1,000 bees on the 24th of July. 0.01 heritable component. Well, that tells me, and should be sending off alarm bells to you, you can't just look at mites on the adults to do your breeding. The reason why is because there's usually about twice as many mites in the brood than there are on the adults. And when those mites are in the brood, you have to know if they're reproductive or not. So any program solely based on the number of mites on adult bees is probably not going to make a lot of progress because of the low heritability component. And we'll come back to that as part of the breeding program for developing varroa-resistant stocks. And so that's just what I don't take my word for it. Take it right directly from John and Jeff. So they said in their manuscript, quote, measuring mites per 1,000 adult bees probably would fail to use any progress. Measuring the final mite population, the growth of mite populations, is marginal and would probably yield very slow progress. Again, a very low heritable component to that measure. Low heritability suggests that progress would be slow in trying to enhance these general characteristics, perhaps because the growth of a mite population is affected by many unrelated characteristics. Let's say you had a really good IPM program, and that's why your mites were very low. There's no heritable component to that. So you have to just breed for those things that have the highest heritability. Breed for the process, not for the outcome. Okay, so when they evaluated those heritable components, they found out that it was all part of this behavior called varroa-sensitive hygiene, or VSH. And James and I will bring that up again later. You don't want VHS bees, you want VSH bees. So it works like this. So as that varroa foundress enters the cell, and it is then capped, and that larvae starts to pupate, at a specific time in that pupil's development, so typically around late wide eye, early purple eye, but not too late. It's not definitely the dark-eyed stage. A worker bee will detect that there is a reproductive mite in that cell. When that reproductive mite is identified, it will uncap the cell, and then they will, and in some instances, they'll remove that infested pupa with the varroa. The varroa will also exit the cell once it's uncapped in some instances. So this is the pattern of varroa-sensitive hygiene. It involves three distinct phases: detection, uncapping, and removal. And we'll see how those are important for developing a breeding program a little bit later. And here's what VSH looks like. So you can see that this bee is uncapping a cell. You could definitely tell if the cell has been recapped or not, and I'll show that a little bit later. But so that's the detection phase and the uncapping. Now you'll notice on this next set here, they are actively removing the infested pupa. And in most situations, they'll either destroy it, you can see destroyed pupa here, or they'll remove it from the colony as part of just the hygienic behavior. And you can see they're recycling the pupae here. But notice here at the top, look at this varroa coming out. So that is enough to interrupt the varroa's reproductive cycle and make her non-reproductive. You can see her jump onto this adult bee here. Okay, so that's VSH in video form. So just to recap here, is that VSH is a behavioral trait. This is not a stock. VSH is a trait within a stock. In what I'm going to talk about later, it's in the stock that we developed here at the lab called Paul line, right? This exists in many other populations, typically at, I think, about a 5 to 10% frequency. Once you can identify it, you can breed from it. So VSH is a trait, not a stock. It results in low mite reproduction, and colonies with high expression of VSH confer strong resistance to varroa. It's different than general hygienic behavior, right? So the hygienic behavior, the freeze-kill brood assay, is really good for the bees to cue into diseased and dead brood. It is not the same as varroa-sensitive hygiene. Varroa-sensitive hygiene is a specialized component of hygienic behavior. And it also looks a little bit different too. So on the left-hand side here, this is what uncapping will look like in a colony with high levels of varroa-sensitive hygiene. You can see here again, I remember I told you that they uncap typically at late white eye, early purple or pink eye to very early purple eye. And you can see there, all those cells that are uncapped are in that pink, that early pink white eye kind of stage. You can notice that. That's going to look a lot different than when you're evaluating hygienic behavior. because when you're doing hygienic behavior with free-scale brood assay, this is what it'll look like. Other thing with general hygienic behavior, they'll remove larvae and pupae of any age as long as it's infected in a way that that signals it for removal. So a little bit different than general hygienic behavior. So if you want to visualize it, VSH is a subset of hygienic behavior. All right, so how do we measure VSH? Well, the methods that we're going to talk about here are kind of a combination of methods developed by John Harbaugh, one of the godfathers of VSH here in the United States, Fanny Mondet, who is the research leader of the French version of our lab, and that we collaborate with often, and Garrett Dodds, who's currently at our lab, who does all of our selective breeding. So what I want to do is, if you plan on doing this on your own, that there's a couple of words of advice. If you're unsure of any measurement, err on the side of caution and mark colonies as susceptible, right? Because once you start identifying your breeders, you want to make sure that they're the good ones. So be very, very picky. Evaluate as many colonies as you possibly can. This is almost like it said, it's between 5 to 10% of colonies. And so you're gonna have to really look. So if you're trying to find something that has a 5% frequency, you need at least 20 colonies to find that one, right? So the more colonies you have, the more that you'll be able to identify. And then once you're going to do your breeding decisions, you're going to only want to breed from the best, right? So make sure you're doing the ones that have the lowest varroa, but also to the other things that you really like. So be picky. Another thing when doing this kind of measurements is that you have to have the right equipment. I'm not that old and my eyes are not as good as they used to be. So having good optics will really help you doing this, help you do this. You basically want to use strong colonies and you want to round up all your materials before you start. It's a really good idea to practice methods before diving in. So typically we evaluate VSH in September, October, usually around here. We might do it a little bit earlier, but it's a really good idea to start in June and July when just to get the methods down, to get the practice, learn how to open up cells and identify them, because it takes a little practice. It's not easy, but it's definitely something that you can do, just as long as you have the right equipment. When doing your evaluations, evaluate them for colonies that you did not apply miticides prior to the evaluation. Typically 60 days since the last miticide application is probably enough, especially if you're looking for it. And you have to keep good records. Garrett, who's our breeder, he has stacks and stacks and stacks of pedigrees going back 15, 20 years. You have to keep good records. You can't just, oh, I'll remember what colony that was the good one. No. Put markers on it, keep good records, number them. You have to just be very, very good at record keeping. I always say that the best thing you could do to be a better beekeeper today is keep better notes. And this program's not really that different. Okay. So when you're measuring varroa resistance, it's really important to keep track of the queen. You want to make sure that the queen that is present from when you do your evaluation to when you're doing your breeding. So like when we do that, we usually mark our queens with a dot of red and enamel paint. That's the way that, and then we usually clip wings just so that way we know it's definitely for sure our queen. Perform varroa washes spring, summer, fall, winter to select colonies with low varroa and low population growth. Now I wouldn't breed from those, right? I wouldn't say that's it and we're done, but it does help you identify which colonies might have the lowest varroa numbers in them. And so this is just a hypothetical situation here, is that you can see here over time, as you measure your varroa populations, you can generate these different growth curves, right? So let's say you had a colony and every couple of weeks when you measured it, the varroa population just grow really big. I wouldn't even evaluate VSH in there. You know you don't have it, right? So I would just throw that one out. Don't even use it. You may want to evaluate VSH if you see these things with low population growth. You can see here, they're growing, but not as rapidly and exponentially as in the susceptible. What you really want to do are find the ones in the red. So these ones that are flat. These are really good candidates to evaluate VSH behavior in. So this is a way that you could start with 100 colonies and start whittling down the pool to evaluate VSH, because this trait evaluation does take some time. All right, so we want to keep track of the queen, and that must be in the colony for 6 weeks, mostly because that will ensure that there's at least 2 brood cycles so that all the bees in the colony are her daughters. So 2 brood cycles in 6 weeks should get you that population that you're trying to actually evaluate. Longer, the better. You have to make sure you're not disturbing the brood nest, and brood nest must be undisturbed for at least 30 days. So don't go into it 2 days ago before you do your formal evaluation. So let it go for a while. Mentioned about the varroa populations. And then you're going to want to evaluate varroa infestation from 1 or 2 brood frames in the colonies with the lowest mite washes. So that way you can kind of get a good accurate representation of what's going on. And you want to look at purple eye pupae or later, okay? And this will make sense a little bit more because it's going to tell us about the varroa reproductive behaviors. And then you want to open up cells all over the frames. So don't just focus in like this one little patch of this big frame, just because you might not be getting all the right ages of the pupae, and you might be focusing on too small of an area. Varroas tend to be distributed in patches. And then you want to open up a lot of cells. So from each frame, you need to evaluate potentially 200 cells. The more cells you open up, the more confident you're going to be in your measurements. John Harbaugh will say you only need to open up 100. Garrett says, well, I'll open up 200 if I don't find any mites. And then Fanny sometimes says 500 cells. So the more the better, but at least 100. Okay, so let's look at varroa reproduction because this is the foundation of varroa sensitive hygiene. So under normal circumstances and unselected stocks, those varroa will infest the larva right before it pupates. And then as that larvae develops into a pupa, the varroa gets in there, lays her eggs, and as that pupa ages, her offspring mate, reproduce, and move out as the bee emerges. So you can see here, there's this, and it has to happen on this very stereotypical time frame. The varroa development is specifically tied to honeybee development. If those 2 things are thrown out of sync, the mites will be non-reproductive. So when you look at the age of the pupae, that should indicate where the mites should be in order to be reproductive or not. And so we're looking at varroa reproduction. These are 2 examples of reproductive mites. You can see here the foundress mite here is showing this dark brick red mite here on the left. And you can see her daughters, one of her daughters here, and then the male here, etc. Then on the one on the right, again, you can see the immature female offspring here in the cell. So this is a reproductive cell. Usually mites will deposit that fecal patch on the upper, so if you have the cell, it's facing up, they'll typically lay it on that north side of the vertical side of the cell. If you start to see these fecal patches on the pupae, don't know why, but that's often associated with a non-reproductive mite. So again, check for that fecal patch. That'll help you orient to reproductive or non-reproductive mites. So let's look at these mites. So we have to learn how to age them and even sex them. So the varroa foundress is typically this sort of thing that we're used to when we think about varroa. It'll be darker in color. Usually they're dark brown or brick red kind of color, and that'll be a little bit different than the daughter mites that you might see in a cell. So the foundress is going to be the darkest one. And so this is just what the varroa female offspring look like. So the immatures are this white colored member you can see here. And then you can see as they age, they go through these molts where they get progressively darker and darker and darker until the mature adult daughter is this kind of light peach, kind of orange sort of color. So you could use color as a way to kind of age the varroa offspring. And that's really important because even though there might be offspring, the age, if it doesn't match up with the honeybee pupal stage, can be indicative of non-reproductive mites, okay? So as they get older, they get darker. You can see here, there's just the subtle differences between the foundress on the left and the daughter on the right. Like I said, the daughters are typically going to be a little bit lighter in color. Varroa males, you probably won't ever see these. I've seen not very many in my life, and I've opened up tens of thousands of cells. And so you can see here on the left, again, the immature males are lighter in color than the adult males on the right-hand side here. Identifying the males in the cell, though, can help you determine if they're reproductive or not. If you have a male, you can see a male, it could be an indication of a reproductive mite if it's at the right stage. If you find a male and the pupae's too old, then it doesn't matter. Okay, but male mites. So it's not that easy, but it's also not that hard to determine the sex of the mites. So on the left here, that's an immature female. And on the right, we have the immature males. And the male mites tend to be a little bit longer than they are wide. You can see they're kind of round, whereas the females are a little bit wider than they are long. And then female mites have shorter legs, whereas the males have these longer, spindly looking legs. It might not seem that obvious now, but once you see it a few times, you can actually tell this pretty easily. It's this, but the biggest thing you're gonna be looking at here is that body shape. Okay. So that's a little bit how to sex varroa mites. Okay. All right, so looking at honeybee reproduction, it's tied with the age of the honeybee pupae. So in the bottom half of this figure here, we have different ages or stages of honeybee pupae. So as you can see here, as honeybee pupae get older, they get darker. You notice their eyes get darker and their exoskeleton also gets darker. Using the eye color and this color of the exoskeleton, you could age them along this, this directional pathway. You can see that here. And you have to age the pupae in order to determine if that varroa mite's reproductive or not. So in the upper hand stage, you'll see that if the mites are reproductive, these are the things you'll see in that cell. So every cell that's reproductive obviously has to have a founder mite, and you'll see a founder varroa. You'll see that in every cell. But then you'll start to see that the male, the first egg that's laid, should be consistent with this purple eye pupae. You see that here. So this male has to be, this immature male has to be here at this stage, along with the immature females. And see, as that pupae gets older, you can see that those varroa age. Okay. So age of the varroa has to match the age of the pupae. That's the take-home message here. So let's say we have a situation like this, where as the pupae ages, the honeybee pupae ages, on the bottom part here, in a normal reproductive cell, this is what we would see. We would see that the foundress lays the egg with the mite, with the adult, the males and the females, and as they get older, they have these sort of stereotypical progressions of behavior of color. Now, if we see something like this, though, where you have a foundress, but that male doesn't appear until this P7, the black and tanny gray wing pads kind of stage of that pupae. Yes, it has a male. However, it's too late for that male to reach reproductive age where he can mate with his sister. So as that pupae emerges, there is no opportunity for mating. That's a non-reproductive mite. So whether it's no offspring or asynchronous offspring, where the age of the mite offspring is not tied to the correct stage of the honeybee pupa, it can be reproductive, non-reproductive. All right, so let's look at this in like a quiz almost. All right, so keep your answers to yourself, but let's look at this. So let's say you open up a cell and you had this purple-eyed pupae, and there was a foundress mite, immature male, immature female. Reproductive, yes or no? In this situation, this is a reproductive mite. Let's say you open up another cell and you find a purple-eyed pupa with only the foundress. Reproductive or not? It's non-reproductive, right? At this stage of the game, that foundress should have laid eggs, and you should see immature males and females. Okay. Let's say you open up a late stage pupa. You notice here that they're very dark, and you see you have two foundresses. Reproductive or not? No. What this situation is, when that honeybee pupa is that old, we should have seen male and female offspring that were also very old. With this situation is here, this is a double foundress effect, neither of which reproduced. All right, let's say we have this stage here where it's a little bit younger, but we have the foundress and 2 mature daughters, reproductive or not. In this situation, it is reproductive, okay? This is what you should expect to see in a reproductive cell. Typically you might see 2, but sometimes you'll see 3 female offspring. So it just depends on that particular foundress. Okay, so dark-eyed pupae here, you see we have the foundress and the 2 offspring, reproductive or not. In this situation, that is consistent with what we expect, right? So it's just a little bit older than that purple eye, but still what we expect based on timing. All right, late stage pre-emergent pupae, and we see a foundress and a single male offspring. This is another example of a non-reproductive mite, because that male has only been around at a very, very late stage of the pupae. So this has no opportunity to reproduce, non-reproductive. It had an offspring, it had a male offspring, but not one that was reproductive, able to participate in reproductive behavior. Okay, look at this one. We have a dark-eyed pupa with 2 foundresses, 2 males, and 2 immature females. Reproductive or not? Reproductive, yes. This is actually a double infestation. This will happen typically late in the season as brood population is declining. There's fewer opportunities for reproduction, so typically you'll get these multiple infestation cells. So yes, you can have 2 or more foundresses. All right, let's say you open up a purple-eyed pupae and there's nothing in it. Reproductive or not? No, you can't have reproductive mites if you don't have any mites. So this is a little bit what you'll be seeing when you're opening up your cells to do your evaluations. Another thing to pay attention to as you're going through this to look at VSH behavior is looking at the cappings. So if that capping is has this shiny underside on, that's a silk from the pupa, that's the original cap. If the cell's been recapped, it'll have this characteristic dull appearance. This is something good to measure as you're evaluating your colonies, because typically you could use this as a measurement of, potentially measurement of VSH. It's usually associated. Higher recapping is usually associated with VSH. Usually, okay? They could recap for other reasons. All right, so then when you're taking all this data, this is kind of what the data sheets look like. So again, this is why it's important to take these notes, because as you're going through this, you have to make sure that what you're evaluating is recorded for your decisions. So you record many things here, like the different cell counts, the brood stages, et cetera, et cetera, et cetera. So that way, when you're going through your evaluations, it might look something like this, where, you know, on this date, I evaluated these colonies, what stock it was, and then total number of cells examined. All right, you want to remember I said at least 100, sometimes over 2. We had 103 recap cells. And then as we looked in the infested cells, we found that this one had an adult foundress, it had an egg, and it had a male offspring and an immature daughter. And this would be a reproductive mite. So this is just an example. We can go over this in much more detail later, but this is the kind of data that you will collect as you go through this to develop these resistant stocks. All right, so then we have to take all that data and make sense of it. We have to make breeding decisions, right? So there's 2 things we need to look at here. It's the proportion of cells with non-reproductive mites. So you could have mites and still have varroa resistant stocks if they are non-reproductive. Non-reproduction is really what you're looking for here, okay? So let's say you open up your colonies and you found that 80% of those mites or more are non-reproductive. That's a good one. If you go down, the less and less you go down, you can see these different tiers are different rankings, right? Another thing that you're looking at too are the number of varroa in those cells. I like to use non-reproduction as a more reliable measurement because you might just by happen chance, not all the time, but you just might be opening up your cells that don't have varroa in them, right? The more you open, the more confident you'll be in that. That's why in this situation, if you find 0 mites, you definitely have to open up 200 cells, because like I said, since mites tend to be distributed in patches, you need to open up lots of cells all over the frame to get this number. But the best ones you could find are 0 mites and 200 cells. You could find 1 mite in 200 cells, even if it's reproductive, and that's still good. But then once you get down lower and lower to these different rankings, it's going to really influence your breeding decisions. All right, so when you look at these two things and you could come up with this VSH colony score, you're really looking for these number 4s, either ones that have all non-reproductive mites or 0 mites in 200. So this is if you're falls in either one of these categories. This is what you want to base your decision on. This is the breeder that you're looking for, okay? The ones that we have at the lab, that's what we breed from. We can find ones that have 0 mites throughout 200-300 cells. You could definitely use these ones in that second, the colony score number 3. You could definitely use those. Those are really getting you on your way. That's almost going to be, it's going to be a 4. Her daughters, if they mate with another one, that's also a 3, they'll be a 4 next year. Now we start to get into a little bit of the gray area here. We get into the VSH colony scores too. When you first start out, this might be the best you could find, the ones that score in this level 2. So 30 to 50%, 49% non-reproductive mites, 2 to 4 mites in the brood, that's a 2. That could be a keeper if that's the best. You really don't want to do any breeding with anything lower than this. So anything with a 1 or a 0, I wouldn't even consider it. You can see here, if you find 8 reproductive mites in 8 cells, you're done with this evaluation. So you don't have to open up 100 if you find that many that quick. So this is definitely takes some getting used to, so that way you can identify the flow here. Okay. Next thing we need to think about too is drone saturation, right? Because the queens are one part of it, right? But it's the drones that really also are the complementary part to this. And this is, to drive this home about drone saturation zones, this is a map of our lab. Our lab is located where the blue star is. And this yellow line is the location of a farm in which we have a lot of our colonies that we do the breeding. It's pretty good because we have a barrier here, the Mississippi River. LSU campus is up to the northeast. And this map needs to be updated because this whole area is now apartment buildings. But what I want to show here is these red dots indicate the location of our apiaries on this point. We typically have about 400 or 500 colonies across these apiaries. The colonies that we identified as 3s and 4s, according to those evaluation, get drone comb. We want to produce as many drones as possible. And especially early in the season, you could really pump out a lot of great drones. So using drone comb will help you get your drones. So this is what we do when we do our open mating. We actually have 2 different lines of this pollen line. We have the open mated version and then the instrumental disseminated version that we are evaluating. So this is a great way to do a lot of mating because you can do open mating. But the way that you really want to get quality mating is with instrumental insemination, because then you can control. You can definitely make sure that that queen that you grafted off of a number 4 breeder is getting semen from a number 4 drone, right? So this is a very complicated technique, and this is something that our lab does regularly, and you will make a lot more progress in your breeding if you can do instrumental inseminations. However, anyone who does it will tell you you can't do thousands of queens like this, right? However, you can produce better, more consistent queens by controlling the genetics precisely. So I will get you there a lot quicker. But this is the thing. You can make up either way you do this, it's going to cost time. Either you do it on the front end with instrumental insemination, or you have to make it on the back end by evaluating many more colonies. All right, so a timeline of events would be something like this to do your evaluations for varroa sensitive hygienic stocks. Is that you want to establish your colonies in May, which they will be soon. And then no miticides. That's kind of the big thing here, because again, remember what I said earlier, you have to make sure that it's not some other factor that's keeping your varroa levels low. You want to make sure it's the genetics. So no miticides. Treatment-free is the way to go here. Can lose more colonies, and it can be disheartening. However, that's the way you're going to make sure you're selecting only the best. I would say do your first varroa wash in June, so that way you can see which ones have good genetics. Do another one in July. And then finally in August, should be enough that way you can get the ones that have the lowest population growth. This is going to help you identify your potential breeders, and then you know, okay, let's not disturb this anymore because September, October is when we're going to start to do our evaluations in these colonies. In September, typically that's when we start doing our inspections here. You look for varroa reproduction, and this is when you're going to identify your breeders. This is when you're going to evaluate them as 4s or 3s, right? And then usually this is when we start prepping them for winter. So we'll add more bees here, and then really start pumping them up with feed, getting them ready for successful overwintering. Nothing's worse than finding a great breeder and then you lose her over winter. And then in February, we'll install a drone comb in those breeder colonies, make sure that most of the drones are what we want. And then March is when we start doing our grafting, insemination, and start the process over again. So this is a lot of work, right? This isn't something that you can just wake up and fall into. Why did we go through all this work? Well, because this works, right? I'm going to skip over these few slides. So we do this because we know that Pauline, the one that we have at the lab here, it's ready for prime time. It has VSH, but it also has these things that beekeepers like: rapid population growth, high honey production, high survivorship, and being very, very gentle. These are great to work with. When we do this selective breeding, I'm just going to skip through this real quick so we can take some questions. We did this big study, and this kind of really demonstrated to me. So we did this study with 171 Paulines and 191 non-selected commercial stocks. And then we found out, look at this, lo and behold, in a commercial beekeeping operation, Pauline has twice as high a survivorship as the unselected commercial stock. So you can see that over time, Pauline survivorship is significantly higher. All right. Well, what's causing the differences in survivorship? Guess what? It's varroa. So when we look at September varroa levels here, you can see here in Pauline has fewer varroa mites in September than the commercial stock. Pauline typically has about 2 mites per 100 bees without any chemical interventions, whereas the commercial stock has a little bit over 8. So 4 times less varroa in Pauline, all things being equal. And how do we know that? And then the other thing that we have to point out here is that there are differences in stock sensitivity to varroa. In Pauline, if they have less than 2 mites per 100 bees, they're fine. But if they have around 4 mites per 100 bees, not very high, right? This is 4 varroa per 100 bees. They're dead. However, in the unselected commercial stock, those with 4 varroa per 100 bees in the commercial stock, they're fine. They have a higher tolerance for varroa in that commercial stock. And then the commercial stock, the ones that had about 10 mites per 100 bees, they're goners, right? So you can see there's different tolerances or different sensitivities to varroa among these stocks. Well, how do we know these colonies died because of varroa? So if we looked at the proportion of colonies that died in each situation, so if we look at the percent colony losses, in Pauline, they were about 30%, in the commercial stock, they were almost 65%. And we look at the number of colonies that died with those high varroa mites. So what proportion of those Pauline colonies had more than 2 mites per 100 bees, and which of the commercial stocks had more than 8 mites per 100 bees? We could see that most of those colonies died because they had high varroa, right? So this is really demonstrating the importance of varroa for honeybee survivorship. Good thing is with this stock is we didn't see any differences in adult bee populations in the real world, so there was no trade-off. They had very comparable honey production, so again, not really much of a trade-off. And so what I wanted to really send you home with today is that bees selected for VSH behavior have reduced colony losses under migratory beekeeping conditions. This is something that can be used in commercial beekeeping operations, and even if you have just backyard bees. They're also going to have less varroa transmitted viruses. I didn't talk about that today, but I'll say that for another time. There's very little trade-offs because they have similar adult bee populations, honey production, and also lower supersedure rates. So with just genetics alone, we could get at these big progress here. I did a lot of this work with people at the USDA and at LSU. You can see them here. And with that, I can take some questions in the remaining time.
Well, thank you, Frank. Appreciate that very much. Very excellent presentation. I may or may not have caught most of that at ABF. I may or may not have been talking to Garrett Slater in the back.
Yeah.
Class clowning. the portion that you guys did where people could come up and actually participate in doing, you know, like a field type assay with some actual live brood frames was fantastic, was a great idea, and everybody seemed to be very, very involved.
Yeah, it's definitely something that we want to get out there more. And so we're going to try to develop and see if we get a grant to get people to come here to learn how to do it in the summertime, so that way you're ready for the fall. Because one of the things is it's a technique we do all day, right? Especially in the fall, we're always evaluating this. So we have the expertise and the infrastructure to do it. Not everyone does. So it'd be good to show people, here's how we do it. When you get back to your place, there's the things that you'll need to get. So, yeah. And
So
so,
Michael Coppola has a question.
yeah.
Harbo claimed to have the first mite-resistant bees in 1995 and then released them to the beekeeping industry in 01. 23 years later, why don't we see much resistance in bees in general? And could selecting for disease resistance be a better path? And is anyone doing that?
Yeah, so the original stock that was released a long time ago, it wasn't ready for prime time. It was definitely very good at keeping the varroa levels low, but it did have the trade-offs of smaller colony size, less productive, because they were so focused on breeding just for VSH that they weren't really well suited for commercial beekeeping operation. Unfortunately, that stuck around even through the development of pollen line, that kind of reputation. So when I was telling people about this, is oh, they're garbage. They're not really good bees for beekeeping. Well, we do studies now with pollen line that show no, there aren't very many trade-offs. So I have data on this. Bob Danka has it, and then also too, we're getting more data this year. I shipped out probably like 500 queens this spring already, just to kind of get at this. So that was kind of, there's been a hesitancy to switch over. Another thing has been availability. Some people that we've been given this material to do the development, they've been doing a great job of it, but it just hasn't been at the kind of industrial scale that we need to supply the beekeeping world. They're very rare. At least good ones are kind of rare because, as James pointed out, some people can sell VHS bees. So I don't know if you want VHS, but no, but for real though, is that there is potentially people selling quote unquote resistant stock that might not even be resistant. So that could be, they could be getting a bad name because of that. However, as we get out more and more data, and we all know that with development of amatraz resistance especially, that we need to find other ways to do this. And we have so much data that show that these things are ready for prime time and we can develop it. And we have the tools to do the selections that we're gonna be working on, that I think we're starting to see that groundswell pick up. So for example, I'm working with James on developing this up in Michigan, but also too, I'm working with the California queen producers. I'm working down here in the Southeast with our queen producers. We're working with other regional groups too, to get this to a point where most of the bees have this trait. We know it can happen because 30 years ago, hygienic behavior was kind of something that Marla Spivak was working on, but through tech transfer programs, most bees nowadays have hygienic behavior. So we know that this can be done as long as there's desire from the industry. And I think that desire is increasing, that I think we'll start seeing that better.
And that example with Marla lends to the idea of the heritability of something like hygienic behavior.
Yeah, for sure. So we know it can be done. So that's kind of what we've been seeing. The other thing is that it's kind of, this trait is a little difficult to control unless you have a sufficient population. I showed that in that along the Mississippi River that we had our breeding yards. I mean, we can really saturate that area with drones. Unless you kind of have that kind of community involvement among a group, or you have a sufficiently large beekeeping operation, keeping those good drone sources out to saturate the population is a challenge. That's why it's always a challenge in that first year, even the second year, to have sufficient drone sources. So the way that this trait works is that, let's say we have a number 4 breeder kind of queen and a 0, right? Let's say the daughters from that 4 are mated to drones of a 0. It'll be somewhere in the middle. It'll be about a 2, maybe a 3. But her daughters, that 2.5, let's call it, her daughters, if they mate again to another 0, the trait's gone. So it can go out of the population pretty quickly if you don't have sufficient drone sources. So I think those are all barriers. But the more that we build up these genetics in the populations, the more it'll be self-sustaining. I definitely see that it could be part of just normal beekeeping in 20 years, hopefully.
Hopefully that would be great, right? That would definitely leave a mark on the industry for positive, for positive.
Yeah.
Back to the Harbo Harris documentation on the original study and the evaluation of the heritability. The question I think was in relation to the chart. How was the heritability of those traits reliably determined? Is that
Yeah,
something you?
so so that's something that they've done in that. I can send that paper over. They just compared the performance of those daughters in those colonies to their parental performance to come up with the heritability scores. It's actually not that hard, and I can send over that paper, and then if anybody else wants, if you want to read it, I can definitely facilitate a discussion on understanding it. It gets a little tricky, but it's definitely, so I'll send over that Harbo paper for anyone who wants to read it.
Yeah, we can post that. If it's published, we could put that link in the video notes too.
Yeah, it's definitely, it's available. We'll get it out there.
Perfect. So this is a question that actually comes up quite often with folks that are investigating, adopting the strategies that you've discussed today. Some of the things like, mite loads and, treating after certain thresholds. Mike asks, if you have a high mite load early, would you treat that specific hive? And that's kind of got a lot of the caveats and the depends and, certain issues that come up with all the variables in play. But if you're starting with a high mite load, would you treat that specific hive? Then what? What do you do?
Yeah. So after that, I'd probably remove it from the population, pinch the queen, or move her to a resource yard where you want to keep her away from your the stocks that you want. And that's how we run it. So in that breeding island that we have, if there's anything that we don't like, it gets moved out. Because sometimes they'll have big colonies and we don't want to just get rid of them. So we have resource yards far at the other way down the road that we move them to. Yeah. So treat them because the bees are still good. I would just treat them and move them out, or try to even requeen it. So that's why whenever we're doing our breeding here, we almost always have extra cells and extra queens. Like we bank tons of queens here in the spring. So that way later in the season, oh, that one's not performing well, put a new one in. It's just luxury we have.
Good strategy. Yeah. Yeah. I always have a crisis of conscience when it comes to moving something out of the selection process, because then I'm like, I could just sell her, list her on Facebook or something, and somebody will pick her up. And then I was like, I feel bad selling her because I know she's not doing what I would want her to be doing, but she might be actually just fine for somebody else because she's still got good brood pattern. She's very productive and gentle, but she's just not suppressing the mites.
Yeah. I mean, that's a good one if someone wants to try other IPM measurements, but you know that they don't have the right genetics for sure. Yeah.
This is a treatment dependent queen. Have at it.
There you go. Perfect. Yeah. Full disclosure. Sure.
Yeah, full disclosure. I'm gonna sell you. She's for sure gonna mite bomb if you don't treat her. Okay, so this also is another good question that comes up. If you have 0 mites and 200 cells, how do you know it's a non-reproductive or colony that just started with a low count and then becomes infested? And he asks, in a few years.
No, that's a great question. And that's why we do these evaluations in September, as we expect mite populations to be at their highest, right? After treatment, after long period of no treatment. So like I said, we typically establish our colonies here in April, March, April. It's different every year. So that way, under normal situations, if there are sufficient mites and there are susceptible genetics, we would expect those colonies. So if we did this test now, we probably wouldn't see very many varroa, because like Cameron said, that brood break, which serves as a varroa control. And typically, when we start our colonies here, we'll do a little oxalic acid just to clean them up a little bit. So if we do this early in the season, we'd probably miss it. That's why we have to do these evaluations in September or October. And we know that those varroa populations should be sufficient because oftentimes we're also running a lot of susceptible stocks here just so we have varroa to do our research. And if we're finding, in those unselected stocks, 30, 40 mites in a couple brood cells, and we go in our VSH colonies and we're finding zeros, that gives us a lot of confidence that the mite population should be at the levels that we expect if they were susceptible.
Yep. A lot of caveats.
Yeah. And so and that's why, but no, that's true though, is that's why I like using non-reproductive as a more reliable measurement to do it. So in fact, if we really want to be strict and we find 0 to 100, we might go to 300. Sometimes, like I said, Fanny says 500. So if you find zeros, the more cells you're going to want to open up just to be sure.
Yeah. Yeah, and you definitely have to have some level of mite pressure to get them to a point where it's easier to find those mites. I dread, and I mean just dread, pulling cells and finding 0 mites in that first 100, because then I'm like, oh man, I got to do another 100, and then
Because
if you
you're the
don't find
100
any
first.
more,
Yeah, yeah, go
yeah,
ahead.
and it's good, it feels good. Oh yeah, no mites, but well, I got to keep looking because you need to find a mite, and finding a reproductive mite is going to give you a much more a solidified base to operate from with that colony. So
Yeah, and why it's really important to open up not just 100 from like one set like this, because we know that the varroa are distributed patchily over the comb. So let's say you got those 100 that just so happened to not have it. That's why both sides of the frame and just kind of randomly all over.
yep, yep, yep. And I always see them in these little circles throughout these various
Yep.
portions on the brood frame. It looks like I got most of the questions that were hanging out there.
Yeah. Yeah, and the whole key to this too is just going treatment free. It's a leap of faith that some people are ready to make, and for completely understandable reasons. We're just so used to applying miticides as our normal. It's part of honeybees treatment, right? You put on supers when they need to. You feed them when they need it. You put on miticides when they need it. So it's just hard to kind of get away from that. And for better or for worse, we don't treat at the lab, especially in the pollen lines, at all. And when I say at all, I mean they don't see a miticide unless it's part of an experimental design. And so I kind of am a treatment-free beekeeper, kind of. But we can rely, if we have a lot of confidence that these genetics that we've incorporated over many years are going to do the job for us.
And you have methods in place that are helping you make those informed decisions, and that's a big bonus. And one question I have is on some of the research that you've done with the stocks you've worked with, are you finding, or are you doing any confirmation, like coming back and doing secondary or maybe even tertiary evaluations on this on the Harbo scale on the stocks, or are you just sticking with his his thesis that just once you score, you only need to score her once?
Yeah, it's score once and one and done kind of thing. Because when we go and do those secondary evaluations, they're very similar. Especially if we do them kind of close by. I mean, sure, in the springtime, they might be different than they are in the fall, but if we're evaluating contemporarily, like, a couple weeks apart, a 0 in September is usually a 0 in October and 0 in November. So it seems to be pretty consistent across that sort of that time period that you would expect to see it. So that's using the Harbo test. The tricky thing with the Harbo test is that we can only really have confidence in it when varroa mites are sufficiently high, typically in the fall, September, October, November. What we really want to do is develop tools that we can do this now, right? So we can go out there and evaluate colonies for VSH in May, which would be great because then we could do 3 or 4 rounds of selection a year instead of 1. So the unhealthy brood odor assay that's developed by Kara Wagner, commonly called the UBO assay, we know that it's a pretty good indicator of VSH, but we don't know if we could use it as a breeding tool for VSH. And that's something that we're collaborating with her on, and she's collaborating with a lot of people, because I think that has a lot of potential to be a really great tool for much more rapid evaluation. Second thing we're also looking at too is the genetics of selection. So we think we know the genes involved here. We have the data. Ariel Avalos, he's the geneticist right around the corner. We have the data, analyzing it or obtaining it. It exists. But obtaining it and analyzing it has been a struggle where we can make so much more progress and we have these markers. So what he wants to do is make essentially develop a test where you look at the genetics of your bees and it says breeder or not breeder. And that's kind of where we want to go for this. So we could use genomics, UBO assay, and how does it compare to the Harbo test? So we're always trying to improve the system.
Did you catch that question in the chat? That was the next one that popped up was the UBO.
Oh, from Bill? Yeah. Have you used UBO compared to Harbo? Yeah, that's something that we're trying to evaluate. So like I said, Karen, so this question got asked me earlier. So I was like, hey, Karen, what's going on with UBO? So she sat down and took me to school for like an hour and a half about all the work that she hasn't published yet on UBO. And it's like, okay, there it is. No further questions, Your Honor, kind of thing when I was done with her. And so it was really, really cool to see that, because some of the data she shows, if you don't have her explaining it to you, might seem a little contradictory. However, when she explained to me how it all kind of works, like, oh, okay, that makes a lot of sense now. And so even when I was asking her some very challenging questions, she had an answer. So like, okay, yeah. So I think that UBO is definitely a good indicator of VSH, because wait, let me think about this so I don't say it incorrectly. You could have I'm not going to say it because I don't want to get it wrong. Invite Kara to your next to one of these things, and she'll tell you exactly.
Yeah,
because it does get a little tricky.
Yeah, it does seem like a promising tool for identifying, the high prevalence of hygiene. I think it was was Stephen Martin from the UK was having a discussion with him about the overlap, and he had pointed out something that we do as Americans with the term of VSH, and they have their own vernacular across the pond, but he had made the point that we often will confuse VSH, or we conflate the term VSH with hygienic behavior, absence of the suppressed mite reproduction. And in a sense, at the heart of it, I believe VSH, originally the term was coined to illustrate the suppressed mite reproduction, whereas the hygienic behavior was also a component of it. So it was kind of like a collaboration of the genes in the honeybees. But now we see a distinction between some bees are hygienic, like the Minnesota, without the mite suppression. And so the term is kind of taking VSH for both sides of the word.
Yeah, so you could have colonies with high levels of hygienic behavior that have very low levels of VSH. So not all hygienic stocks are VSH, but all VSH stocks have hygiene. Okay, that's one distinction. Second thing is that the UBO assay may be a reliable indicator of VSH, what Paul mite has. What we need to do is determine what the UBO score is in other stocks that are not VSH, specifically the Russians, the mite biters, other independent stocks outside of our lineages here from the lab. So that, because you could have populations that are very good at keeping varroa levels low with the brood methods, or it's a lot of the European things that they've been finding. They could probably have a pretty lousy UBO score because that's not what they're using to keep mites low. And so I think that's what Kara's data shows, is that the UBO assay is a good indicator of colonies that have low mites due to VSH.
And I'm a big fan of it, because if you got somebody who's treated their colonies, but they really want to turn that corner in the spring and they want to start moving the direction of selecting for resistant stock in their own apiaries, they can do the UBO and get a good indicator of whether or not they can use this stock to do further testing and start working their way to get off of that chemical treadmill.
Yeah. And like I said, this is something that we know is sustainable. These colonies tend to survive pretty long. Garrett he's like, yeah, this is a 4-year-old queen that we have here. So when beekeeping is very different when you don't have to worry about varroa all the time. I'll tell you that for sure.
Yeah, that's awesome. It's a lot of optimism and a lot of hope on the horizon. Appreciate you as always, and I'm glad that you came and spent some time with us, and you provided us with the presentation. It was really great, and looking forward to talking with you more and definitely catch up some more further.
Yeah, and pass my contact information around if anyone has any extra questions. I'm always happy to answer. So whether it's an email or some other way, that we'll be in touch.
1000%. Thanks a lot, Frank. Thanks for your time. Thank your family for your time. We appreciate you.
All right. Good night, everybody. Have a good night. Bye-bye.